All set to go.
So, hi, this is Nick Makridakis. Basically, I'm a director at AstraZeneca and Big Pharma. Before that, I was a professor at Tulane University and the University of Southern California. I have a PhD in biochemistry, molecular biology, and I'm an expert in genomics. So, long story short, I'm Greek. I came here about 23 years ago, did my bachelor's in Greece, PhD in Southern California at USC. And that's where I became professor, and I did some molecular genetics of prostate cancer. Then moved to Tulane to do DNA repair, cancer genomics. And then moved to Boston to go to a biotech company. And now I'm in AstraZeneca doing pharmaceutical drug development. Can hear me fine. It keeps asking me to unmute, but I'm ignoring this. Hello? Can you guys give me thumbs up? I don't know what's going on here. This computer.
I kind of see two screens.
Yeah,
Have you already started sharing anything? Except your desktop.
I understand why this. So, 1 second. Now you can see more one screen again, right?
We
Correct?
can still see you on screen, not the presentation.
No, I know. So 1 second.
Mm-hmm.
Let's try sharing again. Now you can still see the screen. So 1 second, let me make this smaller. Can you see this? Hello?
Yep. Yes.
So
can you enlarge your screen?
Yeah, I can make it into a slideshow, I believe, since this is PowerPoint. Is that okay?
Yes, much better.
So, Genomics in Health and Disease is the name of the lecture for today. Athena Biopharma is a company I started a couple of years ago, but I'm also working in AstraZeneca as a director. So, what is a genome? A genome is a complete set of instructions for making an organism. Basically, a bunch of DNA sequence that is very specific for each organism. Your body has actually 50 billion cells. Well, every species has different. Humans have about 50 to 100 billion, yes. And each one of these cells has an entire copy of the genome, the same genome, generally speaking. Of course, there is somatic mutations, such as birthmarks. These are somatic mutations. And that's why, we say that you're mosaics. But generally speaking, most of your cells have the same DNA, the same sequence, and the same genome. Now, the DNA that makes the genome is coiled, as you see in this picture. And 1 second, it's popping up. So the DNA makes the genome, as you see in this picture. A, G, C, T are the 4 bases that determine this sequence, repetitive many times. Each one of your genomes about 3 billion nucleotides. Each one of these cells about 3 billion. And, it bundles up itself, as you see in the picture, and it forms these small dots that are actually packaged together by proteins. And they're packed really tightly into a thing that's called a chromosome. And you have a very specific number of chromosomes. Each species has its own. Humans have 46, 2 times 23. So basically you have 2 copies of 23 chromosomes, or frankly, 22 plus X and Y. Basically, 2 copies of 23 chromosomes makes 46, and that's a human. Whereas it's, for example, mouse has 20. Every species has a unique number of chromosomes. The fruit fly has 4. So it just depends on the organism. Now, these chromosomes are in the center of the cell. As you see here, purple, this is the nucleus, and that's where all the chromosomes are. And each one of these cells has a complete copy of your genome. And this is what a genome is, the collection of genes, basically, the collection of DNA. Any questions for this so far? By the way, feel free to interrupt and ask a question. Unmute yourself and ask a question, basically. Now, imagine you're in a party, and there's many cats there. You brought some treats, some cat treats, and when you get there, you notice that, the cats are running everywhere. One cat has a long orange fur, the other one has a gray fur, and the other one has stripes. One has a fluffy tail, one has bushy one, one's a stubby one. Basically, all of these things are controlled by what? Genes or genomes? What do you think? Anybody? Question? Anyone?
Genes.
Huh?
I said genes.
Yep, correct. One gene controls each one of these things. They call traits. It's one of these traits. And sometimes it's actually not one gene, but more than one. There's also some traits that are called continuous traits. Continuous traits are something like height, weight. Why? Because there's a continuity of results, right? 1.6, 1.7 meters, 1.5, etc. You can have anything between. Whereas sometimes it's just a dual thing, right? Blue eyes or green eyes, for example, or limited number of options. So all of these traits are controlled by genes. And that's why the cats have different color and type of fur. Now, I saw this in the news last week. Illumina and the San Diego Zoo are sequencing koala genomes to investigate disease. Now, what do you think about that? Is that a good thing or a bad thing? Anybody want to elaborate?
Well, if koalas are experiencing disease, then sequencing the genome could probably find a way to help the disease. There's also other things that you could do first to do it. And there's the question of, how does the koala feel? Are you being ethically, good to the koala? I don't know. I mean, if it works, then it's good. If it doesn't work, it's a colossal failure and they're all going to get fired.
Right. Interesting. So, but it's interesting that both Illumina is, by the way, a company that started next generation sequencing, so which is important for the genomics. But San Diego Zoo is interestingly involved in this. I don't know why they're doing it, but anybody else has any other, different points of view? Just to hear something else. I mean, my question would be, why koala? I mean, they don't reproduce that much. We like rats and mice in the laboratory because they cost a couple of dollars, and you can breed them. You get tons of mice and discover many things very quickly because they breed a lot and they're cheap and they don't cost that much to feed. So basically, space is not an issue. You don't have to worry about their psychology so much. Koala is a different story. On the other hand, koala is a mammal and much closer to humans than the mouse. So maybe if you find something, I remember that they found some viruses in the koala, and they're hoping to find something that will benefit the koala, but also benefit perhaps human disease. But, it's a stretch. And that's interesting. I'm surprised they do this kind of research. I just want to bring it here. But so in medicine, we use genetic markers and genetic maps to identify disease genes. So this example here with the koala, the question is, how do they translate this information from koala to human? And the answer is they can translate this information because there is homology. Many times, genes are similar in different organisms, especially closer organisms, such as, humans and monkeys, or humans and koala even. It's a stretch, but it's possible. So many times we study animals, we humans study animals to find causality for human disease and cures for human disease, because it's much more difficult to do it in humans. And especially, like I said, with the laboratory animals, we can breed them, we can grow them in large numbers and test different hypotheses, like is this gene or that gene, that causes this disease. And the phenotype many times is similar. So how do we do this kind of stuff? We do it with the help of genetic markers. What is a genetic marker? A genetic marker is a marker that tells us where the gene is. And in this case here, you see the genetic marker of Drosophila, which is a fruit fly. The tiny flies, not the big flies, the flies that go in the wine. This is the Drosophila. And this was the first successful gene map that was produced. And the map shows the relative positions of allelic characteristics of the second Drosophila chromosome. The distance between the gene map or the map units are equal to the percentage of crossover between different alleles. What does that mean? What is a crossover? And if we can go here, you can see a very simple cartoon of what a crossover means. And basically means that, like I said before, we're diploid. Most of us, including koalas and mice, are diploid, meaning that we have two copies of each chromosome. One is coming from your father and one is coming from your mother. And basically, in this cartoon, they're different colors so that we differentiate. So it doesn't matter which chromosome you pick, chromosome 1, chromosome 2, it's basically one copy comes from the father, one from the mother. So when you form your germ cells or your sperm cells, you kind of reduce that diploid into a haploid. But in the process of doing that, there's also crossover. So the crossover is shown here. Okay, I don't know if I can point with a mouse. Can you see my mouse? Yes, right? You can see my mouse. So right, this is the point of the crossover, this X that you see. Basically, this chromosome in mitosis is aligned with this other chromosome because they are similar. The chromosome 1 and chromosome 1, chromosome 2, chromosome 2. And then they form a crossover, as you can see. This crosses over the other chromosome. And then they switch species. So you started with two different copies, maternal and paternal, blue and purple, right? But you end up with 4, blue, purple, and purple, blue, right? Meaning that from the beginning to the end, because there's orientation here. There's a left part and a right part. But if you look at it from the orientation standpoint, there's 4 different copies. Why? Because there's crossover. How often is that? Very often, very common. Why does that matter? Because of this, we can actually predict where the genes are. For example, in this genetic map, you see that these different positions are control different characteristics of the fruit fly. For example, the eye color. For example, the purple color is right here. As you can see, purple eyes. Or green is here. This one is where the antenna, the bushy antenna is. And here is the wild type allele or the common allele, and here's the mutant or the recessive. Mutant doesn't necessarily mean that it's bad. It may just be the recessive. But basically it means that its position is controlled by a gene, a different gene. And this was mapped long time ago. Now, why does that matter? Because these are now phenotypic markers that tells us where genes are. In other words, let's say you have a disease gene and it happens to always correlate with purple eyes, then you know that it's near here. Why is it near here? Near this position. Because it always segregates with purple eyes. Why does that happen? Well, because of chance. If you look at here, crossovers occur at a certain spot. When they occur, the genes that are on this side will stay together, and the genes that are on this side stay together. So the longer the distance between these traits, the more likely that they will cross over. And actually, you can see that in the numbers here, right? 0, 31, 48, etc. Meaning that between this marker and this marker, there's 48% that there will be a crossover. The longer the distance, the more likely it will be a crossover. So even though they're in the same chromosome, if they're further enough, they will cross over. And that helps us identify disease genes, because these markers, tells us, aha, this mutation, this disease, let's say diabetes. Flies don't get diabetes. I'm just giving an example. Diabetes occurs always with the purple eyes. Well, now we know that the diabetes gene is somewhere here. Why is that important? Because it's a long chromosome, 3 billion bases. So if we know that it's near a certain marker, we can identify easier. And that's how it is. Now we go in this area around this purple marker, and we sequence the area to find genes. And we find 3 genes. And now we know one of the 3 actually causes this disease. So this is why we care about genetic markers in medicine and in science, because it allows us, if we know the genome and the genes, it allows us to find disease genes. And if we find them, then we can ask the question, what do they do? And if we understand what they do, we can cure it. So that's the process. And that's why we care about it. Now, sometimes we don't have genetic markers, we have the so-called molecular markers. What are those? Well, look at this marker here is pretty obvious. You can actually see it, in the microscope. You look at the fly and you look at if it has red eyes or not. But this one here is not so obvious. It's actually a marker in the DNA. And so to actually see this kind of marker, you have to do a little bit of molecular biology. You have to isolate the area, the genetic area, cut it with a restriction enzyme, and run it on a gel, which is what this is. This is a gel. A gel is basically some piece of gelatin, like a jello, that we run genetic material. And basically, you put some charge, and the DNA, because negatively charged, it migrates. And the bigger the size, the slower it migrates. So smaller fragments migrate faster, and bigger fragments migrate slower. And that's what you see here. Here's 1,000 bases, and this is 0.9, and this is 0.6. So the 0.6 migrated fast. And now this gives you the size of this. Oh, this one is 0.8, and this one's maybe 1.1. And notice how they're different. So, for example, this could be different individuals, and they have different genotype. Basically, they have some mutations that allow them to run differently. So that's why you see different sizes. And let's say now that this one here is a diabetic mouse. Aha, we have a marker for a diabetic mouse. This is a molecular marker. You have to do a molecular technique to identify it. But these are the kind of things that we do in science to identify disease genes. Why is this important? Because now you can diagnose. You don't need to look at the symptoms. You know that anybody who has this genotype, this particular band that runs like that, has diabetes. So that's excellent. You have a diagnostic tool. B, once you know what the mutation is, maybe you can identify why this mouse gets diabetes. So these are the kind of things that we're interested in, and that's how we use genetics to cure disease, or to at least identify the cause for disease, which is equally important. Any questions so far?
Yes. So you said it takes longer to transfer a fragment of DNA depending on how big it is, right? So
Right.
how long would it take to transfer the entire genome of, let's say, the fruit fly?
Okay, very good question. We cannot do that experiment. It would take too long. It's too big. So what we do is we cut pieces. This one here is only 1 kb. There is this particular gel is called agarose, and there's some other gel materials that are a little bit different and they can run bigger pieces. But to be honest, the biggest size, there's the so-called pulse field gel electrophoresis, which can run maybe 100 kb, 100,000 bases. That's the biggest we can run. More than that, it just doesn't separate well. So it doesn't enter the gel. If it's too big, like a huge chromosome, it does not enter the gel at all. It's too big. It cannot migrate.
So then, like the human genome, which is even bigger, didn't they sequence the entire human genome? So how did they do that?
Absolutely. How did they do that? They broke it into pieces and they sequence each piece independently. And then they map it. Because the pieces were overlapping, they were able to use a computer to actually overlap them and find the overlap. You will see in a bit how it's shown here, actually, in one of the slides. Any other questions? Okay. So, how do we use these tools to do genomics? Well, one of them, you might have heard it, CRISPR. CRISPR gene or genome editing. What is CRISPR? Some people also call it Cas9. Cas9 is one of the CRISPR molecules. It's not the only one. Basically, CRISPR are initials. It stands for clustered, regularly interspersed, short palindromic repeats, CRISPR, in short, CRISPR. What are these? These are basically repeats. The key word is repeats. It's a repeat, and these repeats are hallmark of bacteria defense system that forms the basis for the genome editing technology called CRISPR. Now, what does that mean? Well, basically, bacteria have sort of a primitive immune system to attack viruses that want to attack them. So my bacteria don't have immune system, but they have this kind of primitive immune system called, CRISPR is one of them. There's about 3 or 4 of those. And one of them is CRISPR. And we just use the system to our advantage to cut and paste different sequences and to cure disease. How do we do that? Well, here's an example of how do we do that. CRISPR is basically, like I said, the bacterial system. And in this particular case, 3 different ones. And one of the 3 is this enzyme called Cas9, which you see here in this white kind of background. It's basically a protein, a huge protein called Cas9. And that protein actually uses an RNA molecule, shown here, called guide RNA, to cut DNA. Cut is a slang word. Basically, it's a nuclease. Cas9, CRISPR is a nuclease. It's an enzyme that cuts DNA. What does that mean? Well, and why is CRISPR so important? We have many nucleases. I have used many. We use them to do recombinant DNA technology, basically to cut and paste different pieces of DNA together and create new things. Now, why is CRISPR so popular? CRISPR is popular because it's very accurate. That's the only reason it's popular. There's many enzymes before that can do this, can cut genome and can cut genomic pieces, but they're not so accurate, and they have a recognition sequence that's quite small, 4 bases, 6 bases, etc. These are too small and occur in the genome many times. CRISPR is unique because it's very accurate, and the recognition sequence, as you can see here, is about 20 nucleotides. So because 20 nucleotides, that's a very unique sequence. And now you can cut only one at one specific spot in the whole genome. And that's why it's advantageous. Because now, let's say you have sickle cell disease, which means you have a specific mutation at one spot in your chromosomes, only one spot. Using CRISPR, you can correct that specific spot. How do you do that? Well, CRISPR uses this RNA guide and this 20 nucleotide base pair. So you put this sequence that exactly where the sickle cell disease is, -10 and +10 from that mutation. And now CRISPR goes to the exact spot in your genome and cuts the DNA and makes a double-strand break. Now, generally speaking, double-strand break is bad and the cell will die. But what we do at the same time is we introduce another piece of DNA which corrects the mutation, so that the person now, instead of having the mutation that causes sickle cell, has a wild-type genome, a normal genome. And that's how CRISPR corrects the mutation and the disease. Now, there's different variations of this technique, and actually I'm asking a question about that in the test. But basically, that's the bottom line, and this is how CRISPR works. Any questions? Okay, so ethical issues, many ethical issues. So is editing human DNA or CRISPR by CRISPR or other methods safe? Is it ethical? There was a huge controversy. You may have remembered. I don't know if you remember this, but back in 2018, there was huge news from China about these CRISPR babies. Basically, a Chinese scientist introduced CRISPR to cure, not to cure a disease, but actually to confer an advantage to some people because their parents wanted. What was the advantage? The advantage is that they cannot get HIV infection. Basically, CRISPR, the scientists introduced a mutation to a specific gene that allowed the offsprings, the babies, not to be infected with HIV, because we know that about 1% of people cannot be infected because of a specific genotype. Basically, this scientist took that genotype and introduced it to the babies. And now this made huge news, because it has never been authorized. This was never authorized, meaning no regulatory agency authorized it. The parents authorized it, but that's not good enough. And second, because the safety of CRISPR has not been proven in the clinic. There's actually some newer results, more, that have been approved since then, very recently. There's a couple of companies that got approval, but back then it wasn't. And so that's why it was controversial. Now, I am very aware of what happened with gene therapy, because I was at USC. What happened with gene therapy, at USC, what's the relevance there? The father of gene therapy, Franz Anderson, was actually a professor at USC at the time. So what happened is, gene therapy is similar to CRISPR in the sense that you use a gene to cure disease, but not with CRISPR, with introducing the gene in the patient's genome. And the introduction it happens through an adenovirus. An adenovirus is a very common virus, very powerful virus that unfortunately can integrate in random places in the genome. So basically, we have no idea where it's going to go in, unlike CRISPR, which goes in a specific spot. And what happened is this worked for many times, for many patients, but for one patient, this 18-year-old Jesse Gesslinger, it integrated in the wrong spot and activated the cancer gene, and the patient died from cancer. So although the patient was cured from disease, he actually died from cancer that was generated from the adenovirus that we used to cure him, right? So basically, we got after that, gene therapy was stopped for about 10 years until we figure out how to avoid that. And we found ways to avoid that. For example, you can. What we found was if you transfect two different adenoviruses that contain half the adenovirus genome, and they need to recombine to create an active one, Now this cures the problem, because if you have two of them and one is not good enough to create a problem, then they will never integrate in the same spot, both of them. One will integrate chromosome 3, the other chromosome 7, let's say. And so they will never be close enough together to cause a problem. That's why. This is how we managed to control the problems.
So
Yes.
has that adenovirus two-part treatment thing, has it been like clinically approved, or is it not allowed?
Many times. It has been clinically approved. It's been used today in different diseases, including cancer. Yeah. It's called adeno helper, AAV. Adeno, it's like you use an associated adenovirus, basically a defective one. You have two defective adenoviruses, individually cannot infect. Together, when they cross over, they infect. But because they're two different constructs, they never integrate in the same spot. And that's the answer to that problem. Yes, it has been approved and used many times. So, as for the CRISPR babies, well, I don't know. What do you think? Is it ethical or not? I think if you ask Jennifer Doudna, who's the inventor of CRISPR, she would say that it depends on the case, right? If you have a terminally ill patient, maybe it's okay. Maybe it's okay to risk when you have someone who's dying of cancer. When you have someone who didn't even ask for it, like the case of these babies, they didn't ask to be transfected with something that cures HIV. They didn't care. So what if they don't get HIV in their life? Why would they need this? What if it could cause problems? It turns out that, actually, what we found later on was that these babies are actually increased risk for some other diseases. So, yes, the biggest problem with minors is generally that you cannot ask them. The parent takes responsibility for the result, and that's a problem. It's ethically a problem, and we acknowledge that as scientists. That's why we're very careful with this stuff. How we do this, and that's why there's regulatory agencies, government agencies that regulate the process in general. Now, in China, it's a different story. The guy cut some corners and was able to do it by basically lying. Not telling them exactly what he was doing. And that's how he managed to do it. Once he managed and succeeded, he, of course, published it. And then there was the outcry. Now, what do you guys think? Is that, what if you use this, I don't know, to make a prettier human, or to make them, I don't know, taller? Is that okay? I mean, where do you draw the line? What is acceptable and what is not? I think that's a very good question, right? But generally speaking, would you do that? Do you think that using CRISPR to an unproven technique, by the way, and not generally safe, not proven yet safe, to using CRISPR to protect someone from HIV, is that okay? What do you guys think?
Could you use it to get rid of HIV after the person got HIV, or no?
No. This would be only possible. Basically, people who have this gene called CCR5, it's a receptor, and HIV doesn't like this receptor. So if you have this receptor, this variant of the receptor, it's a specific mutation of the receptor. If you have this mutation, HIV doesn't infect you. Natural humans who have this mutation, they cannot be infected by HIV. So what this guy did is he took that mutation and he put it on the babies. So they cannot be infected, but no, if you already have HIV, this would not protect you. No, it's a preventative. And of course, the biggest problem is we cannot ask the baby, right? We make it first, and then they become adults, and then we tell them, oh, by the way, this is what we did. So, there is ethical issue here, yes. And this is a tough question, and I guess it's individual answer, right? Everybody can answer it differently. But clearly, it's something that you need to think about when you make this, and you have to apply to agencies and get an approval from somebody authorized for this stuff. You can't just basically lie and do it. So now let's move on to something more complex. The genomics of complex disease. What is complex disease? Well, so far, CRISPR and genomics that I mentioned was mostly for monogenic disorders that are caused by one gene, such as sickle cell anemia. This is caused by one single gene, and we know the gene. This has been mapped a long time ago. But most diseases are not like that. Diabetes, which I mentioned, cancer, heart disease, those are multifactorial diseases. They depend on many genes, sometimes 50, sometimes 100, sometimes 1,000 of genes, that many. And it's very difficult to find out which genes and how to cure them. You can understand if something is called by 500 genes, how difficult it will be to cure it, because it's very complicated. So that's why you need genomics in this case. You cannot look at one gene at a time. You have to look at the whole genome to identify that cause of disease and what to do about it. Here's an example. So here's a paper that I published in Oncogene in 2020, Genomics of Prostate Cancer by Racial Ethnic Group. So what I did here is I sequenced all the DNA repair genes in the human genome in about 100 patients. Actually, half of them worked, the rest didn't. And I found 1,433 somatic mutations. What is somatic mutation means? It means that the mutation is specific to the tumor. This patient was not born with it. It's only in the tumor. He doesn't have it in his blood. He doesn't have it in his skin. It's only in the prostate cancer that this mutation exists. How do we know that? Because we sequenced blood DNA and the mutation wasn't there. So now that doesn't prove that the mutation causes the cancer. It just means that it was either created by the cancer or indeed, yes, it probably contributes to the cancer. Okay, but that's what the somatic mutation means. And what you see here in this slide is basically the top is the, so I believe the top is the African Americans and the bottom is the Caucasians. Basically, this study was done in New Orleans, and we have a lot of Black population, and African Americans have different mutations than Caucasians. That's what this tells you. And actually, the mutations, they tend out to have more mutations, a little bit more than Caucasians, but also they have different mutations. And what you see here on the x-axis is the gene. Each one of these that you cannot read because there's so many, it's a gene. And each one of these up here is the number of patients that have mutations. So basically, this gene here is mutated in many patients. And this gene here in very few. But you can see a gradient from the many to the less, right? The most commonly mutated ones are here. The less commonly mutated are here. And those things are not the same between blacks and whites, basically. Very complicated story, indeed. Now, even to demonstrate that further, here it is, the landscape of the top most mutated genes in prostate cancer. Now, we have focused on the top 10 mutated genes. And yes, again, you see it in African Americans in A and Caucasians in B. And again, this is the gene and this is the patient, right? And the same thing here, gene and patient. And you can see it's kind of like a jigsaw puzzle. It's kind of like a mosaic, right? What are these colors? Yellow means non-synonymous, basically means a mutation. It changes the amino acid. Blue means synonymous. It doesn't change the amino acid. See green, green means basically you have both synonymous and non-synonymous mutations. And red means stop codon mutation. These are major, the red ones are major mutations because a stop codon means that the gene is completely deleted. Half the gene is missing. So these are very important mutations. But the point is that you see basically a very, very complicated picture. Each patient has different mutations. Some are changing in amino acid, meaning that they translate to the protein. And some do not, meaning that the mutation is in the DNA, but it doesn't affect the protein. The protein is wild type. But you see how complicated the story is. Each patient has different mutations in different genes, right? Even further, now we're looking at the functional domains. We're taking the top 5 genes and we try to identify function of these mutations. What you see in color here is the top 5 genes in Caucasians and African Americans in panel A and B. But now you're looking at the different colors mean functional groups. For example, this protein up here has 2 functional domains, the green and the red. And you see where you're asking the question, where are those mutations with relative to those functional domains? And the answer is the mutations are everywhere. They're not necessarily in these functional domains. What does that mean? Well, it means 2 things. Either these mutations don't do anything, which is very unlikely, because these mutations don't exist in this patient. They only exist in the tumor. They don't exist in their blood. So they were created in the tumor. And B, possibility B is that actually they do something, but we don't know the function of this domain. And that's the most likely scenario. Basically, this picture tells you that we have a lot to learn about what these mutations do. Now, even here, when the mutation is in the functional domain, like this one here is in this green domain, we cannot guarantee that it would make a difference, that it would actually affect its function, but that's a testable hypothesis. Since we know the function of this domain, we can actually test, does it change the function? And I have done those kind of studies, and most of the times you find that indeed the mutation changes the function, as you expect. And that is very significant, because if you have that kind of knowledge, then you can ask for a cure, right? You can ask, if I change this function this way, can I find something, some drug that can inhibit this mutant? Therefore, if I give this patient this drug, he's going to respond. Yeah, because I know why he got the cancer. So therefore, I can treat it. So this is the importance of this genomic analysis. You find information that you can use to cure the disease. Not always, as you can see, the complexity is very significant. So it's very difficult, yet it's possible. Here, you're looking at the mutations per pathway. So for example, these are different forms of DNA repair: NER, MMR, base excision repair, and homologous recombination. This is, by the way, Ashkenazi Jews have these mutations here. BRCA1, BRCA2 is the breast cancer mutation, very common in Ashkenazi Jews. And you can see this mutation in prostate cancer, very common. So is it a breast cancer or a prostate cancer mutation? It turns out that this gene plays a role in many cancers, not just breast cancer. It was found in breast cancer, that's why it's called BRCA1. But the gene controls many forms of cancer, and it causes prostate cancer and ovarian cancer and other forms of cancer. So bottom line is very complex story again. Again, you're looking at African Americans and Caucasians, and you see that all pathways are affected differently from these racial ethnic groups, from one to the other, but basically all pathways of DNA repair are affected. Why DNA repair is so important in cancer? Because cancer occurs through DNA mutations. So when you mutate a DNA repair gene, you lose the ability to repair the DNA. Therefore, you accumulate more mutations, which means more chance for cancer. That's why. So that's why I focus on the DNA repair, because I knew that this is a very good possibility that these genes are mutated in cancer. Is that clear?
I have a question.
Yeah.
Why the gene can mutate? What is the cause of it?
Very good question. So it's a very complicated story. Many are random, meaning, like I said, your genome has 3 billion bases, 3 billion, right? You have 50 billion cells, 50 billion. You can imagine what it is. 50 billion times 3 billion is the number of nucleotides your DNA polymerase copies every time your cell divides. Your cells divide every time. Yeah, 3 billion. Now, these enzymes are very accurate. They make one mistake every, usually 10 in the 6 to 10 in the 9, meaning that they make one mistake every 1 billion nucleotides. but you still have 3 billion. So they make 3 mistakes every time they copy one cell, and you have 50 billion cells. You can understand you have lots of mutations by accident. So that's one reason, accident. Basically, our enzymes are not good enough. They're not designed to be that accurate. Still, 1 in a billion is pretty good enzyme, but it's not good enough. 2, environment. Tobacco. Actually, here we go. Next slide. So as you can see here, types of mutations. You see T2C, T2G, T2C, T2A. These are DNA, right? Thymine to guanine. DNA has only 4 bases, T, A, G, or C. And you can see that in Caucasians and African Americans, the mutations are different. For example, African Americans have more of this T2C mutation. Caucasians have more of this C2A, right? Or G2T. What is causing G2T? Tobacco. So benzopyrene is very well known to bind guanine and causing a transversion to thymine, G2T mutation. So this is molecular signature mutation for smoking. My guess is that these guys here, a lot of these Caucasian guys here, smoked a lot. And that's why they get prostate cancer. Now, it's very difficult to say why you get a mutation, but we know many things. And the answer is diet, high-fat diet, burnt food, tobacco smoke, hormones, old age are all contributors to mutating our genomes. And yes, cancer. But the story is very complicated. And many times we don't know why. Okay, is that clear? So let's talk about something else in the rest of the time. Basically, these are the type of things you can do in genomics, analyzing the whole genome to find the cause of disease. And in cancer is very important because, like I said, cancer is caused by, well, we already know at least 2,000 genes that cause cancer. So you can imagine how complicated the story is. And, that's 10% of our genome. We only have 20,000 genes. That 10% cause cancer. So, of course, it doesn't mean not all of these are mutated in cancer. Of course not. But like you said, if you saw the picture here, if we go back a couple of slides, here's what I found. 24 to 38 mutations per patient. Each patient had 24 to 40 mutations in these genes. Many, right? So you can understand how complicated the story is. No, we're not talking about thousands, but we're talking about research has shown that the mutations are about 80 per person in cancer. Some cancers have more, some have less. Sometimes it's only 12, sometimes it's 5. But in general, it's less than 100 and more than 50. That's the average, basically. A lot of mutations in many genes. That makes it very complicated because now you have to understand what these 40 mutations together do in the same cell. That is a difficult thing to do, difficult to test. You have to introduce them all in the same cell and ask what happens. It's a difficult, you understand, it's a difficult thing to do. We can do it, but it's difficult. Okay, and of course, then you solve it for one patient, you still have the rest. It's a very complicated story. Now, let's go to something a little bit easier. Agrigenomics. So in agrigenomics, we're interested in things such as breeding, pest and disease research, and genome engineering. I will go through details in the next few slides, but basically, the purpose of breeding is to capture the biodiversity to make better outcomes. Pest and disease research is basically the purpose to control to protect the crops and the livestock from pests and disease. And genome engineering, well, actually, CRISPR. A lot of genome engineering has to do with improving the accuracy of gene editing. yes, in agrogenomics, we use genetic not only in humans, but in plants. So here's some of the things we're going to talk about here. Not too many, but, well, these are agrogenomics applications: whole genome sequencing, structural variant calling, targeted sequencing, RNA sequencing, and complex populations. I want to focus a little bit on the, well, targeted sequencing and the structural variant. What is structural variant calling? You guys might be wondering. Well, so far I talked a lot about variation or mutation, but usually like this: A to G, C to T, basically one nucleotide, one base of DNA changed to another, right? That's what you have here, T to G. One T is changed to one G, just a single one out of the 3 billion, right? But a lot of times the variation is not like that. It's actually the whole structure of the DNA is altered. How? Here's an example. You see this sequence here? Structural variation. What does this mean? You have your genome is full of repeats. It's actually junk. You know that 99% of the human genome is junk, right? It's actually ALU elements and line elements, which are parasites. These are genomic parasites. Basically, there's some bacteria plasmid sequences that got into our genome millions of years ago and populate. And today, most of them are inactive. They don't do anything, but they still occupy about 99% of our genome. 99%. Our genes are only 1%. 99% is junk. Now, it may be junk in terms of function. It's not junk in terms of disease. Some of them are still active and still cause disease. But more interestingly for us, we can use it as variants, as markers. You see how here, where is it? So back here, I told you that you can use molecular markers. Yeah, this one here is actually a G to a T, right? A single mutation can make this difference because now the enzyme cannot cut and the piece is different. The size of the piece is different because the enzyme cannot cut it because it cuts GATC and the mutation made it as GGTC and it cannot cut it now. And you see a different fragment. Well, this variation that you see in one nucleotide here, you can actually have a variant here, except that the variation is actually number of copies. Imagine that you have a 2000 nucleotide sequence that repeats itself hundreds of times, and one person has 50 copies, another has 200 copies, another person has 2000 copies. Imagine how would that look. It's so big, it's 2000 times 2000. So it's a huge piece of DNA that if you stay in it, you can actually see it in the microscope. It's that big. And that's what you see here. This is called structural variation. Basically, it's some repeat that copies itself many times in the genome and varies from person to person. And it happens that some of them can be used, for example, to find better markers for breeding or better plants. So this is what structural variation means. It's a different type of variation, but again, the similar story. We just use this as a marker for something interesting. It could be a better genotype, a plant that cannot be infected by viruses or by disease. It could be a bigger core or whatever. It's an advantageous genotype that correlates with a marker that we can easily screen. That's what it's about. The marker is the important thing. Now, what you see here is a population on the left, and you have different species. And now you're basically taking these and you're sequencing all of them in the pot. And you're finding different pieces of sequence, right? And especially if you have long read options like these ones here, you get like 2 kb here, 5 kb there, 1 kb there, and then you map them together and you see some overlap and you say, aha, this piece here maps with this one, and this one here is here. And then you create a map, a genetic map, and you find different haplotypes. What are haplotypes? Well, basically, it's the simple traits that linked together, right? It's like this here. If we go back here in the simple crossover, right? The blue and the purple. This is a haplotype. Now here you have 4 different haplotypes. You have 2 maternal and paternal, and because of crossover, you got 2 more. This is a haplotype because basically it's a string of differences that go together. Why they go together? Because they're genetically linked. They're in the same chromosome nearby each other, so they migrate together. And this is the kind of thing, that's the so-called haplotype. And haplotypes are very important because they determine, for example, a species that can breed, has better breeding abilities. And these are or species that cannot be infected by disease. And these are the kind of things that we're interested in agrigenomics applications. You see, we take this population, we sequence it, and you see now how sequencing works. This is how they found the human genome. The small pieces that they paste together into a whole chromosome. And it was still not perfect because they didn't have the so-called the difficult repeats, like the telomeres at the end, etc. But they had 99% of it. And that's what we usually get. You know, if it's very repetitive, it's difficult to sequence, so you may not get it. But you get 99% of it because that one is not repetitive and it's easy to sequence. And at the end of the day, you get all these haplotypes that you see these different colors, right? And you make a haplotype database. In this case, you say, okay, I understand now that this species has 6 different haplotypes for chromosome 4, another 7 for chromosome 7, etc., etc. And now you can use these haplotypes to find markers that you can use for different applications, such as evolution, to see which species came from which one. We can do that the same with COVID-19, and we have, by the way. We publish these maps all the time. We can do this to find out what variant of COVID-19 is prevalent right now. Haplotype mapping, it's quite simple. And, or what type of virus infects a particular plant. Is it a new virus? Is it a copy of something that we have seen before, etc.? And you can do comparative genomics, meaning that you compare different genomes from different organisms. Evolutionary biology cares a lot about this. Also, comparative genomics tells you how things evolve with time. And all of these things are useful in crops and livestock health. For example, you can use the same approach with whole genome sequencing to monitor invasive species. How do you know that it's an invasive species? Well, you have to do the whole genome analysis, and you genetically identify without any question when you do whole genome, what exactly species is this one? We do this kind of stuff with the flu, by the way, all the time, right? You guys know that this year there's this variant, and that's how they make the vaccine. They know which variant of the flu is prevalent around the world, and then they make this year's vaccine. And that's exactly the same thing that we can do with plants. A little bit different, though. We don't vaccinate the plants. But we understand the pathogens. For example, this beetle that, let's say, infests this plant, we can understand, is it a dangerous one or not a dangerous one? Is there an invasive species that came from another country? Where did it came from? Well, we have a database, so we can just sequence the whole genome and find out what exactly this beetle is, where it came from. So these are the kind of applications in crop and livestock health. RNA sequencing is more specific, right? Instead of the whole genome, which is billions of bases, you only focus on the piece of the genome that makes RNA. Well, that is 1%, like I said, for humans. And for other species, it may vary, but again, it's a small percentage, 5%, 3%, 7%, etc. Right? The majority of the genome is not RNA, it's DNA. The RNA is a minority. It's the piece that makes the genes that make RNA. And then you can use this to understand the host pathogen adaptation, like how does the plant respond to this pathogen? does the plant change with time? Again, you sequence the genome of the plants with time, and you see how they change to adapt to the infection, right? So you can genotype the plant, you can sequence the genome of the plant as well as the pathogen to understand that interaction. And targeted sequencing, that is when you have something more specific. Let's say you want to identify a gene that resists disease. You found a genotype, you found a specific subspecies of this corn, let's say, that is resistant to disease, and you want to find out exactly what is the gene. Well, that's when you have to use first your markers to find the chromosome, and then you narrow down to the specific gene by doing targeted sequencing, meaning that you PCR amplify only specific region of this gene. And let's say that region has maybe 10 genes, and you find which one of these is variant, right? Which one of these, and how do you do that? Well, with molecular analysis like this here, exactly like this. You find finally a region that maps with the genotype of interest. For example, let's say that this one here is resistant and this one is not. Well, now you know where is the gene, because this region is very short, it's like 1,000 nucleotides, so you know exactly where it is. And then you map the gene, you sequence it, and you find out, okay, what does this gene do and why is it resistant? And after you understand that, you know why it's resistant and you know what to look for in the future. And now you create more copies of this one because it's resistant in the future. It's resistant to disease. It's very desirable. And this is the kind of stuff that, that you do to improve crops and to prevent disease. And I'm not going to talk about metagenomes. That's a little bit too much for you guys, maybe. So CRISPR, last slide here. So what you see here is application of CRISPR in agrogenomics. And especially what you see here is how the length of the sequence. All right, let's first explain what this is. What you see from the left to the right is basically a sequence, let's say for a gene, a genetic region. And this bar here, you see this line? This line is exactly where CRISPR cut the DNA and insert the piece. So for each one of these, a different insertion of CRISPR, right? Let's say you want to insert at a specific spot. What ends up happening is the so-called off-target effects. Although CRISPR is very accurate, more than 99% accurate, there's still about 0.1% inaccuracy, meaning that 1 out of 1,000 times, it goes in the wrong spot. And that's what you see here, because although the majority of them are like this, 100% homology left and right of this insertion, as you go down here, you see variants that are not. You see that it's not homologous here. This bar is homology. This 100%, this is 0. So you can see that sometimes it inserts in a region that is not supposed to insert. You basically target the wrong chromosome, the wrong spot. And that's happened. That's exactly why we're saying CRISPR is not safe. Because although it's 99% accurate, that's still not good enough if you change in the genome, right? Because once you change the genome, this is a heritable change, meaning that it will always be there. If you change the first cell, right? If you change it with CRISPR and you make the mistake, that mistake will be inherited to all the cells in that person. They all will be mutant. So that's why we're saying, you have to have a backup. Like we had with adenovirus, where we had a version that you can correct, you can avoid that potential mistake, right? So these are the kind of things that you want to do. And this genomic analysis allows you to see in agriculture or in human disease, where do CRISPR inserts its DNA? What type of mistakes happen here? Why did this happen, right? Why do you have, basically, it appears it has a last deletion. Why it happened? Most likely because the DNA repair mechanism recognized this as a problem and removed the piece of DNA that we want to insert, and actually removed even more, probably a couple of thousand nucleotides if you notice the size of this deletion. So this is what it appears to be the case here, that basically this kind of analysis tells you not only how accurate you are in your technology, how can you make a better CRISPR? For example, let's say we make a mutation on this Cas9 that becomes more accurate. That would be great, right? You would be able to see it with this kind of method, because you would sequence the results and say, aha, I only get this one, this like 100% insertion exactly where I want. So this mutant CRISPR is a better one. So these are the kind of things we do to actually, improve the technology. Any questions? About CRISPR or genome engineering? And I guess this is the bottom line. When used responsibly, genome manipulation analysis can help identify the cause of disease and improve our lives. But again, responsible use means scientific learning and regulatory interactions. Basically, you have to apply to the government to tell them what you're doing. They have to review it and agree before you proceed. And then you do clinical trials to test it. And once it's safe, then you can give it to more people. That's what we do. And by the way, I'm doing a couple of phase 1 trials right now, which means safety. Phase 1 is safety. Basically, you prove the safety of the drug before you go to the next step, which is to prove efficacy. That will be step 2, phase 2. Any questions?
Yeah, it's just a minor question.
Uh-huh.
So, you work at AstraZeneca, right?
Yep.
So in the top right corner, it says Athena Biopharmaceuticals. Is that
Yes.
is that related in any way to AthenaNet, like the healthcare or whatever?
No, it's my own company. Since I'm Greek and I was born in Athens, I call it Athena. But because they had already Athena Help and AthenaNet and Athena, I used Athena Biopharma because it didn't exist. So I got it the proprietary name. That's it.
Oh, that's cool.
There's no other reason. Yeah, so AstraZeneca is not related to Athena. Athena is my own company.
Hi. Sorry.
Yes.
I
Go
just had
ahead.
a small question. I came a little late, so maybe you said this before. I may have missed it, but do you think there's a chance that genome manipulation will be regulated? Because how you were saying it before, how when used responsibly, it can identify the cause of diseases, it can help diseases, it can improve our lives. However, gene manipulation can not only change around the genes of plants to help identify diseases and stuff. Maybe I'm getting it wrong. It can also change traits, possibly in humans as well. And if this isn't used responsibly, it has to be regulated somehow. So what do you think are like the statistics of this thing getting regulated? And
So say it again. What's the
sorry,
question?
I was saying that what do you think are the chances of this thing getting regulated? Because when it comes to gene manipulation, not only could it be used on plants and stuff, it could also be used on humans. And gene manipulation can not only help treat diseases and identify diseases, it can also change traits. And
Absolutely. That's why there's a process, right? So when we use it there's a difference between research and clinical trial. Like research, I have done many things in the lab that are okay. Cutting a gene and changing a lipid, some bacteria is okay in the lab. However, when you go to apply something to humans, it is illegal to do so without proving that it's safe first. And that means you apply to a regulatory agency, usually the FDA. In the US, it's the FDA. In Europe, it's the EMA, etc. So AstraZeneca like I said, I'm in 2 trials. These are international trials. One is in New Zealand, one is in England. They're all over the place. We apply to regulatory agency everywhere, and we prove. The trial usually takes. You guys may not know this, but very briefly, every drug takes about 10 years to be approved. 10 years. Every trial is about 2 years. So the actual trial may be only 1 year, but then you have to, or 6 months, but then you have to analyze the data, understand it, and then submit to the second step, like phase 2, let's say, again to another regulatory agency. This is a process that takes a long time. And only after these 10 years, you get some approval. That's how it is. And that's how it will be, because you're right, it is dangerous, and we don't want to hurt people. We want to improve things and make them, cure them, not hurt them. So that's a process that we have to do. That's why it's so expensive. I mean, people say, "Hi, drugs are expensive." Of course, they are expensive. But do you understand that every drug that fails costs us $1 billion, right? We lost $1 billion. Nobody knows about it, but we did. Only 1 out of 10 drugs maybe makes it to the end. The rest 9 do not. We still spend $1 billion for each one of them. We lost $1 billion. So when we have one that works, we actually make the money for all of these 10 that fail. Do you understand? This is how it goes. This is the reality. And again, I work for big pharma. I can tell you this because that's what I do. Right? I know how it works.
Just another thing, right? When I say what's the likelihood of getting this thing regulated, what I mean by that is okay, so an example of this is it is AI, right? Which I'm bad at communicating my thoughts. So AI has a lot of really good benefits, but a lot of people are wondering if the use of AI, the overuse of it by people, are actually ruining some industries when it's trying to help, right? Such as the music and arts industry. What I'm trying to say is, it's not difficult when something is clinically, when something is approved, it's gone through the 10 years process, it's not difficult for someone else to take this research that's been used and use it for something unethical.
Well, okay, so that is a different story. You know that a drug has some patent rights, right? So we only use things that are patented. And we have about 10 to 15 years patents. Patents work for 10 to 15 years. After that, it's basically, it expires, and then anybody can use it. Having said that, it's quite difficult in most cases. The time that, there was aspirin passed. Small drugs is not what makes money anymore. Nowadays, what makes money in pharmaceuticals is actually the biologics, such as the one I'm working on siRNA, for example, one of my drugs. I'm working on most of the drugs I work with is biologics, and those are difficult to make. Why? Because first of all, the sequence is proprietary. Nobody knows it, the exact sequence. Understand? It's not published. So we say, okay, we're making half of this and half of that, but the exact sequence matters, and we don't publish that. So it's difficult to copycat. A.
Okay.
B, I cannot promise that everything's like that. We still have some small molecules. There's still a possibility that somebody can copy it and use it once the patent expires in 15 years. Yes, it happens. AI is a little bit different. I don't want to elaborate with that, but it's not a software, and biologics are different things, right? Software is much easier to make and imitate rather than a biologic drug. Why? Because biology is very complex, and that's why it takes 10 years to get approved, because there's many unknown things that can happen. For example, we had some drug that we were working with, and it has different effects in humans than in monkeys. We tested in monkeys, we tested in animals. They didn't show that effect. The drug actually works better in humans, but we didn't expect that. And not only we didn't expect that, but it turns out that we have to, extend the clinical trials just to follow up these patients because the drug works for longer. So there's always things that can happen in a trial. That's why it's a long process and it's a complicated story. And yes, we do care about that, and the ethics and the appropriate use. Yes, companies are for profit, absolutely. They want to make money, but we want to make money while benefiting the patient. It's not like just make money for money. That's our point of view. And yes, we know that it's going to take time and we're going to lose a lot of money in the process because most drugs don't work.
Okay, thank you.
Mm-hmm. Anything else? Let's see. So to have. Okay, so let me just. Shasta, are you still around?
Yes, I'm still here.
I guess we're a little bit over time. So there is some questions that I had there, but I guess you guys will send me. The questions are written. Some of them are very easy to answer, and some are more complicated. So I don't know how you guys want to do that. If you want to collaborate in groups and answer those questions, I don't know what Victoria wants to do. I haven't done this lecture before, but I wrote them differently. Like I said, some are multiple choice, they're very easy, basic stuff that anybody can answer in 2 minutes. And others are a little bit more difficult. You have to go to the literature and do some searches. And by the way, feel free to use anything you want in this lecture or Google it, and come up with answers that you think are reasonable. Whether you do that individually or you do like 3 guys together and another 3, it's up to you guys. I don't know. Yeah, I just want to see that you guys do some work and you write something unique. Either 3 together or each person, right? You see my point.
Okay. So would you like to stop sharing?
Yep, I will stop sharing. Thank you guys and take care. Talk to you later. I'll see you again in when I see the results.